Yes, we are live. Doing the last checkers. Okay. Looks like we're live, and we can get started then. Hello everyone. Welcome to our fourth lecture. This week we're going to be mainly describing the data structures and algorithms that are used to identify the similarities between the reads and the reference genomes so that we can facilitate a quick search in the large space of the reference genomes. So if you remember, last week we started essentially with, we described the sequencing technologies, and then we mentioned that all of these sequencing technologies that we covered share a common disadvantage, which is the reads that we generate, they lack the order and the location information regarding their origin from their corresponding genomes. In other words, we don't know where these reads are coming from in their organism because they don't carry that location information and also order information. Rather, we are essentially stuck with, let's say, this bunch of random reads, let's say DNA fragments that may have either short or long lengths. And the goal essentially, or the problem that we need to tackle in that case, is we need to reconstruct the entire genome from many sequenced reads if our goal is to analyze the entire genome. And we said that this is like solving a puzzle. The reads would be the pieces of the puzzle, and then we would need to essentially reconstruct the puzzle from these reads or the pieces. And one good news was that we have a reference genome that we can look at so that we can take some hints, let's say, from the reference genome to identify the corresponding position of these reads. And there were essentially some trade-offs between certain technologies. For example, some technologies produce short reads, making reconstruction of that puzzle more challenging because we may not be able to figure out whether a piece may be coming from this, for example, this position or this position, because the pieces would be too short, making the identification also challenging, accurate identification challenging. For long reads, it's a bit easier, but then long reads are essentially a bit more erroneous. So this is around now 5% to 1% rather than 15%. And essentially this figure was showing the trade-off between the short reads and the long reads, depending on their lengths and then their accuracy. And this essentially is the pipeline that we are interested in solving, in covering throughout our lectures. And we already covered the sequencing part, and we said we would be focusing on this other step in the genome analysis, which is the read mapping step, that tries to solve that puzzle. So the read mapping, in a general term, the goal is to solve, let's say, life's puzzle from sequencing output. So we're not really solving the real puzzle from pictures, but in a sense, we're trying to solve the life's puzzle, which is DNA or RNA, let's say. And there are essentially several ways to, two main ways to perform read mapping, but the most common way is to map the reads to a reference genome. The reference genome is, let's say, a previously known and representative sequence of a particular genome, particular organism. Let's say it's previously constructed. We mentioned the human reference genome from the previous lectures. So we may assume that that particular reference genome exists regardless of how accurate it could be or how complete it could be, which we also discussed in the previous lectures. So to map the reads to a reference genome, so this is how a DNA fragment logically would look like, and this is how physically may look like. And the reads that we're generating are essentially some random fragments from that genome, from that DNA. And the goal is to essentially figure out where these reads belong to in their corresponding reference genome, right? So we first need to find their corresponding positions, their candidate regions in the reference genome, which in this case, for this read, this could be somewhere in this region. And the next is to align the reads to the reference genome. In a sense, now we want to identify the exact matches, mismatches, insertions, and deletions compared to that reference genome, so that we can identify the genomic variations that the individual possess compared to the reference genome, which is a representative of some, let's say, some certain population. The challenge here is that when we try to map the reads to reference genome, then this means that we're trying to map the billions of short fragments of DNA from lengths to 50 to a few kilobases, let's say. But there can be millions to billions of such reads that we need to map. So, and the human reference genome is also large if we are mapping the reads to a human. So this means that like there is essentially there needs to be some efficient search that makes it practical in this large space of the reference genomes, right? So this is essentially another way of doing read mapping. I mentioned two main ways in the previous slide. One way would be mapping the reads to a reference genome, and the second way would be mapping the reads without the reference genome, right? So we may not have a reference genome for every organism, but in that case, what are we going to do? So we can still do or generate, reconstruct the genome again, solve the puzzle somehow without looking at the picture. And the way to do it is to look at the reads, look at the essentially the similarities between reads, and then try to somehow identify matching similarities between them, so that we can reconstruct the puzzle by looking at how similar each piece to each other is. And so this is essentially what we get after sequencing. So these are reads, and they have different, let's say, portions of the sequences. And what we can do is we can find overlaps here, right? So this part may be overlapping with this part, and this part may be overlapping with this part, and so on. So the colors indicate this shared content between reads. And of course, we don't know which strand is being sequenced at the time. So this means that maybe the reverse complement of the DNA will match with the forward version of the DNA, let's say. So this means that we need to take the reverse complements somehow. So I do the animation again here. We take the reverse complements of the sequences to figure out whether there is a matching region of these reverse complements with the other sequences. But then what we do is we somehow remove some unnecessary or extra information, and then find the link between these overlaps, so that we can take somehow consensus to construct the assembly from these overlapping pieces. So this is a high-level explanation of how construction of, let's say, a reference genome, or as we call a de novo assembly, is performed by looking at the overlaps. So we're going to be covering this step more in the next week. And so if you go back to a read mapping to a reference genome, so we said the reference genome is huge and the read is relatively shorter. than the reference genome. So you may assume that this is the entire reference genome, let's say, and this is the read. And one way to perform a search to identify whether this read, let's say, aligns to that reference genome, and to identify how it aligns and where it aligns, what we can do is, starting from the very first position of the reference genome, we could essentially try to align every character of that read to that region in the reference genome. And then we could essentially do this for starting from every character of the reference genome until the end of the reference genome in this way, essentially. Like this read would be traversed through the entire reference genome, performing the same alignment operation over and over again. But this is very expensive. And imagine that you need to do it for every read, that we may have few thousands, millions, or billions of reads, depending on the data set. But then the question is, will it be practical? And the answer is easily no. And you can guess why, looking at this complexity. It would take perhaps years to complete such an alignment for a single data set, given a regular von Neumann architecture. Then there should be some ways, right, that makes this read mapping step practical and more efficient. And of course, the answer is yes, we're going to be covering these. And such an analysis is usually done in 3 steps. So we're going to be covering these steps, but before that, I also want to show how we sequence, how we store the sequence data to, let's say, common file formats to store the sequences. One format is called the FASTA format, is over here, and the other one is FASTQ. But these are pretty similar to each other. In the FASTA format, we have two lines per each read. So a FASTA file may include many reads, right? And for every read, we have 2 lines. Or let's say 2, so it's not necessarily 2 lines, but let's say we have 2 layers for every read. So one layer is an identifier, is an identification layer or identifier line, let's say. And this identification gives a unique, is supposed to give a unique name for that read so that we can identify that read. For example, when we map this read to a reference genome, we want to basically look at this FASTA file again and then see what was the sequence, right? Maybe the alignment information will just include this ID information. It will say this ID maps to a reference genome at this position, but optionally, it may not include the exact sequence information, and you may want to go back and see what sort of sequence it was. So in order to essentially extract the sequence information accurately, we need to have an ID to go and look at. Excuse me. And in the second layer, so I said incorrectly, sorry. So I said 2 lines, but it's not essentially a line because there are multiple conventions of FASTA file. So you may, for some formats, you may see 2 lines, really. One line is the ID, the other line is the full sequence. But some other formats, you may see essentially multiple lines for the sequence and 1 line for the ID. And that multiple line may include the fixed length sequences, except the last line. But regardless, what we have in FASTA is the read ID starting with this particular unique character greater than sign. And then until you see the next read ID, or maybe until you reach the end of the file, whatever you have in between will be the read that corresponds to that read ID. So I hope this was clear. And the FASTQ is basically very similar to FASTA. Additionally, it includes some quality information per base. Here, these are encoded with the ASCII format, and then each number, let's say that each character is corresponding to shows the quality of that particular character. So E, although I don't remember on top of my head what was the exact value for E, but essentially by looking at this, the corresponding values of these ASCII characters, you can essentially see how accurate this base is. And these quality scores are usually assigned by the base callers. These base callers translate the raw data to the bases to indicate whether this base is likely to be to a sequencing error or it is an accurate, let's say, a base. So this is providing some additional information about the accuracy of each base, these FASTQ files. And that's why we have Q here is a quality. So then I guess we can start covering how we can perform this quick or practical read mapping in 3 steps. So what we really want to do is that we don't want to check every character in the FASTA file and every character in the reference genome. Rather, somehow maybe take some subsequences, maybe even sample subsequences in the sequence file, maybe in FASTA or FASTQ. And what we want to do is that we want to quickly somehow identify whether they exist in the reference genome or not. And if they exist, maybe we can go further and do more complex operation rather than checking every character for every position in the reference genome. Perhaps this could provide a more practical solution. And this is what we're going to explain in 3 steps. So the step 1 is usually is an offline step, is usually a one-time task where we extract some subsequences from the reference genome. So you may assume these different colors are different subsequences that we extract from the reference genome. And somehow we want to store these subsequences using some data structure, such that when we query this data structure with some other subsequence, what we want to achieve is that we want to quickly be able to say that whether that given subsequence exists in the reference genome or not. So this means that we need to extract subsequences accurately and then store them such that we, in an efficient way, we can query this data structure efficiently to answer the question whether a given subsequence exists in the reference genome or not. And if it exists, maybe we can say, oh, then maybe there is some similarity over there between the read and the reference genome. So such a data structure, one way of implementing such a data structure is to implement hash tables. There are other ways of doing it. For example, FM index is perhaps another way of, let's say, indexing the reference genome, but we're going to be covering the hash table based indexing in this lecture. So the way that it works is basically what we do is we extract the subsequences. Sorry again. So we extract the subsequences from the reference genome. So assume that the green is a certain subsequence. And what we do is that we use this subsequence as a key in the hash table. So there are multiple ways of converting this subsequence to a key. We could simply directly use the sequence itself, the string itself, as a key, right? But then there could be some problems because that subsequence could be, let's say, extremely large, maybe, let's say, 50 character, 100 character long. But this means that you need to really have a huge hash table to, let's say, reserve the efficient space for all such possible keys. So then I guess one other efficient way of doing it is to somehow hash these strings and then generate hash values from these substrings, so that we can use these hash values as a key in the hash table, which we refer as also a seed that are stored in the hash table. So then what we store additionally is that we store the value for that corresponding key, right? So in the hash table structure, we have a key value pair. Given a key, hash table returns a value if it exists. So in our case, our value is the list of locations that particular seed exists in the reference genome. So imagine that I give you, let's say I give you the yellow seed, right? So I query this yellow seed in the hash table. Then what it will return me quickly is that what it will tell me is that this yellow seed exists in these positions, for example, 3, 5, 12. So when I look at these positions, maybe I'll see directly the yellow, right? So here the third positions somehow, and the fifth position somehow. So this is somehow a quick way of identifying whether a subsequence exists in the reference genome and where they exist, if so, right? So this can quickly tell me whether the particular subsequence is located in the reference genome. So these are essentially seed locations located in the reference genome. So these seeds are usually generated from, as I said, subsequences, which we refer to them as k-mers usually. So that k-mer literally means a string of length k, right? K is corresponding to the length of the subsequence. For example, if I tell you a 20-mer, then this means that we are extracting some subsequence of length 20. And of course, depending on, I guess, the courses that you've taken previously, or the applications or the projects that you work on previously, you may have heard of this k-mer in different terminologies, maybe an n-gram or q-gram. So these are all referring to the same thing, essentially. But then the question is, which seeds or k-mers we need to store in the hash table, right? Because depending on which seeds that we're storing, this significantly affects the accuracy and the space requirements of the hash table. And there are essentially different strategies to select and store the k-mers. So one strategy is to extract all possible k-mers from a reference genome. So we call it extracting all overlapping k-mers. So they are overlapping because of the reasons that I'm going to explain next. So imagine that this is our reference genome. It starts with GCTA and then it goes on with some other sequences. If you want to extract all overlapping k-mer, then we need to start from the very beginning. And there should be a length to that k. So in this case, the length of the k, or the length of the subsequence that we're extracting, is 7. So we extracted the first k-mer. So the next k-mer would be essentially the next subsequence that we would extract after shifting this, let's say, window by 1 character, right? So we were at here initially, now we're shifting by 1 character, and now we're here. So this is what we have now. So this is the second k-mer. And then we extract the other k-mers by shifting again 1 character, so on until we reach the end of the reference genome. So this means that now we extracted all the overlapping k-mers. So here, the k-mer length is 7 again, because the length of the subsequences are 7. And what we usually do is that we hash each of these subsequences or k-mers using some hash function, usually a low collision hash function, and then store the hash value as a key, and store the position of this k-mer as a value in the hash table. So it's like a list of positions. So if I see this particular k-mer somewhere else, then this means that I'm going to generate the same hash value. Then I'm going to have multiple positions for that k-mer as a value in the hash table. So this means that I'll immediately know how many locations that this particular k-mer appears in the reference genome. So if in case I need to go to each of these positions and then check to do further computation, let's say. So the benefits of such an approach is that it has high sensitivity because there is no information is lost. Where literally, maybe there is even a redundant information here because subsequent k-mers contain some redundant information, right? So here you can see this type of sequence is shared across k-mers. So this means that no information is lost and there is even redundant information. And this means that as a downside, it also incurs large storage space requirements. So these are essentially the benefits and the downsides of storing all k-mers in a hash table. So the question is, do we have other strategies that may provide perhaps slightly less accuracy, but still maybe a better storage requirements? And the answer is yes, for sure. One strategy for this is called essentially minimizers. And this is basically a sampling strategy. And how it works is as follows. So imagine that we extract all overlapping k-mers again. So we're not storing them yet, but still we're extracting them, right? So these are all overlapping k-mers. And then at the time, let's say we consider only a W amount of these overlapping k-mers. In this case, W corresponds to a window, and in this case, our window is 4. We are somehow considering 4 k-mers at this point, and the length of these k's are 7. So what we do is that in a given window of k-mers, we hash all of these k-mers in the same window, and then generate their hash values. So what we do is that we find the hash value, we find the minimum hash value. In this case, maybe this one provides the minimum hash value, and then store only this one in the hash table. Rather than storing all of these, we store the one that provides the minimum hash value, essentially, in the hash table. So doing this, we avoid somehow storing all of these in the hash table, and rather we store only the minimum hash value. So this means that we sampled almost 1:4 ratio, let's say. So this means that then our sampling strategy, will it make the mechanism less accurate? So so the answer is somehow yes and no. First, let's mention the benefit of this approach. As we said, there's a reduced storage requirement due to some clever sampling, right? But the downside is we have a reduced sensitivity, of course, compared to extracting all k-mers, because definitely we're losing some information. But the question is, how much of information are we losing? So if you're interested in this, there's a paper that describes the theoretical, let's say, proof behind these minimizers from 2002. Unfortunately, I don't have the link here. But I guess if you search for the minimizers and 2002 paper, probably you're going to identify that paper. But essentially what that paper tells us that if 2 documents or 2 sequences are similar, let's say, so in this case, 2 sequences may be some region in the reference genome and a read, right? Assuming that this region and the read are similar to each other. So this means that if you set your window parameter and the k-mer parameter accurately, then this means that it is guaranteed that they will share a minimizer. So then there's some theoretical proof behind it, that 2 similar sequences, given that these parameters are set right, 2 similar sequences will share a minimizer. So then this is a good approach, a good enough approach, right? Maybe it's not reducing the sensitivity that much. So then we're sampling it still, and we're not reducing accuracy significantly. So at this point, I'll give a 1-minute short break, and I'll continue quickly afterwards. Okay, I guess we can continue. Sorry about that. I guess my throat is somehow blocking me to essentially continue smoothly. So I needed to get some water. All right, then let's continue. So the other approach is called space seeds. So if you look closely to the previous approach, what we're essentially doing is that we are hashing some sequence and then storing in a hash table. So this means that when we find the match for a hash value, so this means that we found some exact match for that particular sequence. So that exact match requirement is somehow limiting some of the applications, because if two sequences are similar to each other, they don't have to contain, let's say, exact matches, and they don't necessarily have to contain exact matches, right? They may still contain some few mismatches between them, but still those sequences could be similar to each other, right? So then this means that by requiring this exact matches, maybe we are somehow missing some important locations or some important matches that we could find, let's say. And the idea behind spaces is to somehow minimize such an effect, so that we could identify, let's say, matches between similar k-mers. And the way to do it is to apply a pattern to the sequences before hashing them. So it's in a sense that we are inserting some don't care characters at a fixed positions of the k-mers, right? So assume that our pattern, for every sequence, our pattern will insert don't care character at the second, let's say, and the fifth positions of any given sequence, right? So when you do this, then this means that if we have, let's say, mismatches at these positions, right? Then if you didn't apply a pattern, then this means that you would generate completely different hash values, because this is how most of the low collision hash functions work, right? So they don't care about the similarity, but rather given some sequence, it will generate a hash value. And regardless whether two sequences are similar or not, these hash values will generate a completely different hash value. So this means that even if you have one, for example, mismatch between two sequences, then you may end up getting completely different hash value. Then this means that you may not be able to find matches between them. But then if you insert these don't care characters, and if you are lucky such that your don't care characters appear at the positions where the mismatches appear between sequences, then this means that you just made these mismatching characters same, because you inserted don't care character here, for example, an X. So this means that when you hash them, they will end up generating the same hash value. So again, let's look at here. If you just hash these, they are different sequences, right? Because this character and at this character, they are different. So they should generate different hash values if you use a low collision hash function. But when you apply don't care characters here, then this sequence is exactly same as this sequence somehow. And this means that if you give the sequence to a hash function, then it's supposed to generate the same hash value. Then this means that you'll find matches, although the original sequences were somehow they were not exact matching. So this is somehow improves the accuracy, right? This is the benefit of this approach. But then one downside is that there is a poor flexibility here, because those patterns are usually a fixed pattern. This means that they are applying don't care characters at a fixed position, and then you should get lucky, let's say, to essentially be able to generate the same hash value for similar sequences only if the mismatches appear at these don't care positions. So this is essentially the trade-off or the downside of the benefits of using space seeds. And this is good for, let's say, tolerating mismatches. And there is another seeding approach. We call these approaches seeding algorithms, by the way, or k-mer selection algorithms, because they tell us, they tell us the mechanisms, how to select the seeds, and these seeds are stored, let's say, in hash tables as hash values, right? Essentially, whatever the hash value that we're generating here is a seed, and seed to the hash table, and the mechanism that leads to that hash value is a seeding technique, to explain it broadly. And another seeding technique is a stromers. So this is somehow similar to minimizers, but it takes a further step, and there are several ways of doing stromers, and this is one of relatively the easier mechanisms of stromers. So what it does is that, assume you have these sequences, and these sequences may be similar to each other, but only maybe they may still include some different inserted or deleted characters between them. But in general, maybe they could be similar to each other, like these long sequences. So if you want to then somehow tolerate these insertions and deletions that may happen between similar sequences, what we could do is we could first identify the minimizers, let's say, within each sequence, and then consider only these minimizers, and in a sense that link them, right? So this means that you somehow ignore whatever in between these minimizers, and then just link these minimizers together as a single sequence. So what happens is that if two sequences, let's say, share a series of minimizers, then they should end up generating exactly the same stromer sequence, right? So we call these stromer sequences or stromer seeds, because if they share the same series of minimizers, if we link them, then they'll end up generating exactly the same sequence. So this means that when you hash these, then they are also supposed to generate the same hash value. So this is then like enabling or finding multiple minimizer matches at once, let's say, between sequences, rather than looking at them by one. And this provides certain benefits, especially when you consider not only we can apply minimizer technique here, but maybe some other techniques, some random techniques, let's say, that may define which characters to link here, such that once linked, perhaps maybe two similar sequences will have the same stromer sequence, eventually the same hash value. So this is another seeding technique. And the benefit is, again, the increased sensitivity because it tolerates the insertions and deletions. But downside is, again, there is some reduced flexibility because the selected k-mers, once linked, after linked, they should exactly match between sequences. So I guess, like, you can see these seeding approaches, even though they tell us how to generate the seeds and how to generate the hash values, they still require exact matches of the resulting, let's say, sequences, right? And this is still something that we want to avoid. We still want to, even though we generate the seed sequence, and these seed sequence may be, let's say, may not be exactly matching with some other seed sequence, but maybe it includes only one mismatch or two mismatches. If you look at here, for example, these two stopmers are now exactly matching, but they may not be exactly matching, for example, if there was a single character change here, right? But then the question is, does this mean that these two corresponding sequences are too dissimilar to each other? In most cases, the answer should be no, because if there is a single mismatch, they are potentially still similar to each other. But since essentially how we're using these hash values, because we're using local origin hash functions, even though there's a single mismatch over here, the resulting hash value will be significantly different. So the question is, how can we avoid this? So there is another seeding technique that is referred as fuzzy seeding, fuzzy seed matching. And the goal is to essentially tackle that problem. So we identify this fuzzy seeding mechanism as like, it can enable certain things. For example, it can enable assigning the same hash value to highly similar seeds. So remember this space seed, right? So these are, although we applied some pattern here, these two sequences are not exactly matching, right? Because here there's still a mismatch. So then this means that you were just unlucky selecting this particular pattern. So if you had selected this pattern, maybe you would end up generating the same hash rate. But then the question is, maybe here there's also some other mismatching characters and so on. So this is a never-ending question. So then this should be whatever the sequence we have, whatever the sequence we generate after applying the seeding mechanism, if that resulting sequence is highly similar to each other still, regardless it is exactly matching or not, a fuzzy seeding technique should enable generating the same hash value so that we can find matches with a single hash value lookup, matches of these highly similar seeds, right? By looking at the hash table. And still, if two seeds are highly dissimilar to each other, such a fuzzy seeding technique should still generate different hash values. They are too different than each other, right? So that we don't find matches between highly dissimilar seeds. And of course, such a single hash value lookup can be high performed because we are still using hash values to query the hash table. So this is like a single query to the hash table, and also the space efficiency because we're storing the hash values in a compact hash table and so on. So then this means that if you are using low collision hash functions, then this means that you need to find exact matching seeds. And this means that maybe you're missing some useful and novel seed matches by doing so. And for such a fuzzy seeding technique does not necessarily use low collision hash functions, but maybe uses some clever techniques to be able to generate the same hash value for highly similar seeds. And so that it also enables rethinking the other parameters related to read mapping, etc. Further. And this is what we do in this paper. This is a blend that was published early this year. This is a mechanism that can enable generating the same hash value for highly similar seeds. And if you're interested, you can take a look at this paper to learn more about it and how we do it, how we enable fuzzy seed matching. But essentially, depending on the mechanism, the seeding mechanism that we're applying, this is essentially defining the content of the hash table, or let's say the index that we are building. And it is also defining how quickly the indexing technique and also the seeding techniques are defining how quickly we're building that index table and how large it is, etc. Right? So then there are different indexing techniques. They have different trade-offs in terms of their, let's say, seeding time, meaning how long it takes to query that index to identify the seed matches, right, and how large the index is, etc. So I'm not going to go into too much detail, but you can essentially check this paper to learn more about it. So this is the first step. It was quite long, but we'll go over the rest of the steps relatively quickly, at least the second step, because I believe you'll understand the gist of it more easily now that we covered the content of the hash table nicely, right? So when you're building the index using hash table, the keys are the seeds, essentially the hash values of certain subsequences that we generate from reference genomes. And the values that we store along with these keys are the list of positions that these subsequences exist in the reference genome. And this is a one-time task. I'm saying this is a one-time task because we do it for every reference genome once, let's say, assuming that the reference genome stays the same, which is not always the case, but the same reference genome is used multiple times, let's say, right? So then this means that you can index it once and then use it multiple times. And of course, depending on the parameters of the seeding technique, you may need to reconstruct the index structure again, but we're assuming that given a parameter and given a reference genome, that is constructed only once. So then the next question is, how can we query that index? In our case, how can we query that hash table? so that we can find similarities between a read and the reference genome quickly. And this is the second step, querying the index using using the seeds from reads. So what we do is we apply the same seeding strategy on the reads again. For example, if we had applied minimizer seeding given certain parameters such as W, window, and K, we should use the same parameters here so that we'll find consistent matches. So we apply the same seeding on the reads, and this means that we're extracting some subsequences again from the reads. And what we do is that we use the same hashing mechanism again to generate the hash values, and then we query the hash table that we constructed previously and ask whether that particular seed exists in the reference genome. And if it exists, then I quickly can tell that, okay, then there is the shared subsequence between a read and the reference genome, right? Quickly. So you remember the very first naive approach that I showed you. In that case, we would need to be checking every character one by one in the reference genome and every character in the read to identify whether there is a match and whether that read aligns well to reference genome or not. But here, what we do is that first we start with some subsequences that are extracted from the reads and also from the reference genome. And then first we try to find quick the matches between these subsequences before we do further analysis, right? So this enables us to reduce, let's say, the space, search space dramatically from the entire reference genome to the certain positions in the reference genome that this hash table tells us. So the hash table is telling us, given that subsequence from the read, you need to go and look at these positions in the reference genome, and then that's on it, let's say. So this is more or less the abstracted way of it. And then we do this, right? We take the seed, let's say, and then it tells us these positions, and then we essentially go and check the other seed, and then it tells us these positions and so on. So the other step that we do is that the seeds that we extract from the read and the locations that we extract from the reference genome, is there essentially a list of positions that are close to each other, right? Because then if there's a list of positions or the chain of seeds that are close to each other in the reference genome, then this means that the order of the seeds also, and also the distance of the seeds also matching the distance and order of the seeds in the reference genome. So it's not like only blindly matching or finding matches of certain subsequences, but then also the next step could be checking. Now you have, let's say, multiple matches of seeds, right, between the read and the reference genome using different seeds. Is there essentially a chain of seeds, or chain of different seeds that appear closely, both in the reference genome and the read, with a similar order that they appear in the read and the reference genome? And we call this a chaining technique. That's also a dynamic programming technique that I'm not going to explain here, but chaining algorithm can give you this so that you can somehow filter out some of the seed positions quickly and then focus on some other ones that are essentially close to each other. Or there are some other filtering techniques that quickly filters out some unlikely positions. Because it's important to do so, because if we still apply aligning the algorithm for every position that we get from the reference genome, still it may be too costly. Maybe there are some obvious positions that we can quickly eliminate before doing the alignment, so that again, it won't be a costly search algorithm. So there are some heuristic and optimizations over here, as you can see. So essentially, querying the hash table with hash values of seeds, it enables us quickly finding a list of possible making locations. So this was the second step. And the third step, so assume that now we have some, let's say, regions that we identified as similar between the reference genome and the read, thanks to, let's say, some chains of seed matches, let's say, right? And we can say, starting from this position of the reference genome to this position of the reference genome, there may be some similarity for that particular read, starting at this position and ending at that position. So then the question is to finalize that question, right? So how can we tell quite confidently that the read and the reference genome at that given position is highly similar to each other, meaning they differ from each other by certain edit operations, right? So the edit operation, what the edit operation would do is when you apply that certain edits to one of the pair of the sequences here, this could be reference genome, the portion in the reference genome, and this could be a portion in the read. And what the edits could tell us that, for example, if you change this and change this, meaning when you make two edits, then this sequence will exactly be the same as this sequence. So the goal is to identify that, how many edits that, and what sort of edits we should do so that we can make this sequence identical to this sequence, right? And this is what alignment does. This is a dynamic programming type of approach. It checks all possibilities in a sense for every base given a region, and then tries to tell you that whether that particular pair of sequence aligns to each other. And the alignment is defined by, let's say, the scores or the penalties, let's say, that are assigned depending on the matches or insertions, deletions that each base may have at each position, which can then finalize the alignment answer. So I quickly mentioned the edit distance or the edits that we may have between a pair of sequences. And edit distance is basically the minimum number of edits, let's say, for example, insertions, deletions, or substitutions, needed to make the, let's say, one sequence exactly the same as the other sequence. And in that case, our sequences are reads and the reference segment. So in this case, for example, we have organization and operation sequences, let's say, and maybe assume that organization is the reference sequence and the operation is the read sequence. And when you apply an alignment, you can get such an operation. For example, here, the blue may tell us there's an insertion, right, in the read compared to the reference genome. Here, the yellow are the matching positions. Here, this pinkish part is the deletions, meaning there are some deleted characters in the read compared to reference genome and so on, right? So all of these operations, when you count them, you're going to count 7 edits, let's say. And this is essentially the optimal, minimum number of, let's say, operations that you can do to make this operation string identical to the organization string. And there is, as you can see, there is no one answer. There could be multiple answers to do so. But what we're interested in mainly also, what is the minimum number of operations? And given that minimum number of operations, can we tell that these strings align to each other? Of course, these are all set by these scores that fill up the dynamic programming table that I mentioned in the previous sample. And this is another example, again, that tells us these two strings align to each other given operations. So since alignment algorithm is very costly because it requires filling up this dynamic programming table for every segment, every pair of sequences where one sequence is coming from a certain region in the reference genome and this other one is coming from a certain region in the read. And these regions are defined by, let's say, seed matches and other optimizations that we apply next. Our goal is really to apply the alignment as few as possible and only for the true regions, let's say. So we really don't want to apply alignment for regions that are not going to end up aligning. So if we do so, then this means that we just wasted computation because we aligned these two strings for no reason because they ended up not aligning to each other. So we wasted a lot of time and energy, let's say, and we really want to minimize this. And there are algorithms that are filtering algorithms and the chaining algorithms that tries to essentially reduce the workload of alignment in the previous step so that we only apply alignments as few as possible for only true regions. And still, even though there are certain optimizations, we still see that the alignment takes up still the large amount of time in the existing read mappers because how costly it is. so there are essentially certain challenges that you may have already observed so far in read mapping. One challenge is we really need to find many mappings from each read, because a single read may be mapping to different regions in the reference genome, right? So then how can we find all such mappings efficiently? There can be differences between a read and the reference genome. So this means that we don't really want to basically rely on exact matches between reference genome and the read, right? Because an individual may have mutations compared to reference genome, because we all are individuals, and due to the evolutionary differences, due to the somatic mutations, etc., our genomes contain differences compared to every other person out there, right? So we want to identify these differences so that we can analyze the genome and make it more personal to you. And that's why we want to tolerate essentially the mismatches between a read and a reference genome. And this means that we need to apply some alignment algorithms to do so, which is costly. And we need to do them really fast. So read alignment is costly because it is a quadratic time dynamic programming algorithm, because we fill up the table, and also it has data dependencies. That means that you cannot really, if you have the budget for parallelism, but still you may be limited by the algorithm, right? Because of the data dependencies, you need to look at some previous cells to calculate a particular cell. Then this means that this particular cell is dependent on the computation of these previous cells. So this means that you cannot really fill up all the tables at once in parallel. So you need to wait for that dependency to be fulfilled. And of course, almost the entire matrix should be computed. There are some optimizations, such as the banded approach, that may avoid computing the entire matrix, but in most cases, we're still filling up some large space of the entire matrix still, which is requiring still a large amount of space and computational complexity. And if you're interested in learning more about, let's say, why this is very costly to do so, you can check this paper that provides mathematical proof to it. Also, we have another paper that describes all these steps that I mentioned you and how they evolved over the last 30 years in this survey paper. So if you're interested in, you can check this one as well. And this is a paper that includes the steps from sequencing all the way to variant calling. So if you want to basically learn more, you can check this paper as well. So I guess the other question to answer is perhaps, why do we even have a single reference genome, right? So far what we mentioned is that there's a read, and then that read maps to a reference genome, and that reference genome is defined as the representative, let's say, sequence of a particular organism. But this is essentially what it is, right? So the reference genome, for example, for a human reference genome, contains around 3 billion characters, and it determines certain characters of an individual, right? For example, eye color, the shape of face, their allergies, and other things, etc. But essentially, how it is generated is basically from, it's not representative of the entire population of human species, right? It's only a representative of certain individuals, and it may differ from individual to individual, but we're still looking at this particular reference genome to identify the differences from reads compared to that particular reference genome, right? And we may have millions of reads and the origins are not known. And what we do is that we essentially find some exact matches between this reference genome and the reads that we have, and then try to reconstruct the rest of the face, the rest of the picture, starting from these exact matches, let's say. And maybe up to a certain point, we may be able to construct this picture of an individual by looking at the reference genome. But the problem arises, let's say, substantially different than the reference genome, such that it incurs variance, right? Because the reference genome and the individual will not be exactly the same. It will include some variance. So this means that we may still be able to map our reads and align our reads to that particular reference genome because they will share enough similarity, let's say, right? Because we use alignment algorithms, and those alignment algorithms tolerate mismatches and insertions, etc. So they tolerate differences up to a certain point. But how about the reads that even the alignment algorithms cannot align? So it doesn't mean that these reads do not belong to that individual. It may still belong to that individual, but we may not simply be able to align it because our algorithms cannot find sufficient similarity between the reference genome and the reads. And this is just simply because we use that particular reference genome. So if we had used, let's say, a reference genome that is relatively close to that particular individual, then maybe we would be able to align these reads to that reference genome using the same algorithm. So the question is using a single reference genome a good idea? And this is basically known as the reference bias. And to solve this, there could be two perhaps main approaches, right? One approach is then let's not use single reference genome, but let's construct multiple reference genomes and map the reads to each of them one by one. Right. So this means that this is an approach maybe that can improve the accuracy, but this is not a really scalable approach because we're literally increasing the runtime by n times if you use n different reference genomes eventually. So then this is not relatively a scalable approach, essentially, right? So this is, for example, we may have 4 different reference genomes. Then the question should be map our reads to each of them one by one. So the other approach is maybe, so the key idea here is that although these reference genomes are still going to be different than each other, in most regions, they are still going to share some similarity as well. So why don't we leverage that key idea, right? For example, the Thai region over here in this picture is similar across multiple pictures. Then the question is, do we need that redundancy in different reference genomes? Or can we somehow merge all these shared information and still keep the differences also in order, so that we somehow be able to analyze all of these differences and still spend less time on the common regions, right, between multiple reference genomes? And this is basically the idea in using graphs when mapping the reads to a reference genome. So what we can do is that we can construct a single graph where the common regions are merged, let's say, into a single node, if you want to already start imagining the graph. And for the variations, we have, let's say, divergence from those nodes, right? Maybe one divergence may lead to another node showing the difference of a particular reference genome, and another node may be showing the difference of another reference genome at the same position, let's say. So this means that the differences can be encoded nicely as well as the similarities. And this is perhaps one way of imagining it. So maybe here the differences are here, but perhaps for one reference genome, we may have, after ACG, we may have T. For another reference genome, we may have G, right? And in this way, we didn't store ACG multiple times, but only once, because it's a shared attribute apparently across reference genomes. But now we were able to also now store differences, let's say, across reference genomes using such a graph structure. So essentially, it's also a good time to think about the graph type of approaches and how we map the reads to a reference genome. But we're not going to cover it in more detail now in this lecture. But we will have essentially a talk that is from our group and where we will explain how we perform and accelerate such a read to graph mapping, let's say. So with that, this is the end of the lecture, and let me give you some teasers for next week. So for next week, we're going to go into the details of the de novo assembly construction. We're going to answer the question, what happens if there is no available reference genome of an algorithm? And if you remember, I showed you if there's no reference genome, we can still find overlaps to construct the assembly of the reference genome of an individual. And this is what the de novo genome assembly is doing. It's building some sort of graph, and then from that graph, it's trying to identify the genome itself. So we're going to cover these steps. And also, we're going to briefly be covering the steps in variant calling that essentially uses the alignment information in reads and tries to correctly identify the mutations in an individual compared to reference genome. So with that, this is the end of the lecture. So I see one question on YouTube. The question is this an AI thing? So since I'm not sure at which point that Lucy asked this question, so I cannot perhaps accurately answer this question well, but almost none of the steps that I explained includes, let's say, the AI operations. But the question is, why not, I guess, right? So maybe there are some opportunities over there to utilize some AI operations. So in the variant calling step that I showed previously, here, there is an ML approach over there to identify the mutations, which I'm going to mention next week. But for the previous steps, so far at least, we haven't mentioned any AI-related approach. I hope this was at least a broad answer to your question. So I assume there is no other question on YouTube. Maybe I can wait a few more seconds just to see if there's a follow-up. If not, I will end the lecture. Okay, I don't see any follow-up, so maybe it's a good time to end. Thanks, everyone, and I'll see you next week.